Well, thank you for inviting me, and thank you, Jerry. I want to acknowledge Jerry's formative role in my own career. Sometime in the late 1970s, that long ago, I was a hapless Harvard medical student wandering aimlessly in the halls, and Jerry took me in and, in all seriousness, helped strengthen my motivation to enter this broad field of neuroscience relevant to serious disorders, of which autism is one. The problem is that we really didn't have any scientific toehold in whether it be autism or other severe neuropsychiatric disorders like schizophrenia. We had sometimes wonderful, although not always agreed upon, careful clinical descriptions. But from the point of view of rationally advancing diagnosis, I guess I could use my level here and not hold it, of rationally advancing diagnosis and moving toward therapies, it was, is this? It was very much a featureless landscape. And, and I think the complexity was certainly part of it. When I was in the beginning in the mid 1990s, the director of the NIMH and very much wanted to push this field toward better understanding and therapies. Once again, the lack of really deep scientific leads made it very difficult to attract outstanding scientists into the field. And I must say that when I left NIMH in 2001, I was feeling rather hopeless. Now, we scientists love to think that it is our ideas that move science above all. But actually, tools and technologies are very important. And what has really turned the tide is our ability to do new things in ways. So I have some agency in this. Now, how's that? Okay. Tools and technologies have really turned the tide. But I always think in terms of Galileo. Now, Galileo was, of course, a brilliant scientist. But it was really incremental advances in optics that, and he built his own telescopes, of course, but it was incremental advances in optics that allowed him to see the first 4 moons of Jupiter and come up with a new conception of the solar system and other things like getting excommunicated and so forth. But without that tool, right, it would have been impossible. So from the point of view of understanding autism and other neuropsychiatric disorders, a number of marvelous things have happened, and they've happened within the same decade. And this has allowed us, I think, as a community, to go forward. So first is genomic technologies. Now let me just say right here, you're going to hear my starting, from the small end of things, and Kathy Lord starting from whole human beings. But of course, the goal isn't to have some reductionist picture of humanity, but rather to exploit the clues that are hidden in our genomes. Because our genes, in the end, encode the RNAs and the proteins that are the building blocks of our cells, and have everything to do with how neurons will eventually function. And insofar as autism is highly genetically influenced, and some other diseases are, the purpose of finding genes is really to get clues to biology. And we'll come back to that. But at any rate, the tools and technologies that were so critical are genomics. So I think we're beginning to understand that autism, bipolar disorder, schizophrenia are highly heterogeneous in their etiologies, but tend to converge in the brain through some final common pathways that we desperately want to understand. But this genetic heterogeneity is enormous. And while there are individual mutations that can act alone to cause syndromal autism, that is core social deficits, but also often intellectual disability and epilepsy, and we'll come back to this if there's time, much of, I think, the core social disability and narrowed interest and repetitive behaviors is probably highly polygenic in most people with autism. And when I say polygenic, I mean maybe it's a little bit ahead in schizophrenia, and we can already see that it will be many hundreds of genes, perhaps 1,000. Now, we have about 20,000 genes. How is it possible to have so many genes involved in a particular disorder? And the answer is we use a lot of the information in our genomes to build our brains and maintain them. It's true that severe protein-destroying mutations can really damage brain development severely and send a person off on a path of disability. But for many people, there are just these tiny nudges, just the little tiny signals, tiny nudges in the genomic and epigenetic and so instructions to build a brain that lead to something like, we think, the picture of social cognition and so forth that we see in autism, or the picture that we see in schizophrenia. Now, when you have many hundreds of genes contributing relatively small effects, it becomes, I'm not going to go into it, but you can imagine, how do you pick these things out? Because the human beings have enormous background variability. So the question is, how do I find a variant that is relevant to autism against this enormous background variation? Well, where does this background variation come from? So human beings in around 1850, at the time of the Industrial Revolution, numbered something less than 500 million people on the planet. A very small number of generations later, right, in 2000, we hit about 7 billion humans. And we know that with every meiosis, with every baby born, there's a new 50 or 60 mutations introduced. But when we become an exploding population in such a short time, in literally 15 generations, there's no time for natural selection to have any impact on which genes are being carried forward. And so there's, the human genomes are enormously variable. In order to pick out the genes that are, or the variants in genes that slightly incrementally increase the risk of autism or schizophrenia, it becomes an enormous numbers problem. So the technology, the ability to sequence DNA has come down more than 1 million-fold in the last 10 years. And the ability to use microarrays to genotype people is just amazing. I mean, we're working with Illumina and a consortium to create what's called a psych chip, and it'll be updated every few years. And it's going to have a really good array to understand somebody's genetics in general, but then every variant of every kind, copy number variants, damaging mutations, and SNPs that have come out of all of neuropsychiatric research, and we'll be able to, for $65 a person, genotype everybody. And I think at a time like that, it's then the genotype-phenotype relationships will, I think, begin to open up in a way that we've all been looking forward to. But it's the ability to study genes cheaply, rapidly, and accurately that has allowed enormous progress to begin, and it's only going to get better. Now, a second technology that's really critical is stem cell technology, I believe. So why is this? Well, the tradition, if you have a single gene disorder one gene has carries all the weight, has a big biological effect. The traditional approach has been to make a transgenic mouse, to put the human disease gene into the mouse, and then study what goes wrong in the mouse. Now, there are several problems with this traditional approach. And by the way, it's been done and produced very interesting results with some mutations that also produce syndromal autism, SHANK3 and the Fragile X mutation and so forth. The problem is if you have many hundreds of genes, making a mouse and breeding it and having enough is a year's project. It's very expensive. And we need high throughput systems to be able to interrogate a large number of genetic variants that are contributing. The second thing is that many of the variants that we see, and we don't know this for autism yet, but I'd be surprised if it isn't true. It is certainly true for schizophrenia and bipolar disorder, are not in the protein coding regions, but in the regions of the DNA that decide when a gene is on, where it's on, for how long it's on, how much it's on, these regulatory regions. And this kind of regulation, especially in the brain, is barely conserved at all between humans and rodents. And so there are certain risk variants that you simply, even if you could afford to make them mice, it wouldn't be informative. So the second technology, which is amazing, is the ability, and look, it's early, we're not there yet, but we can take skin fibroblasts from people with autism, from people with any other illness, from healthy people, and turn them readily into neurons. We can turn them now into forebrain neurons. We can make genetic inhibitory neurons. We can't yet make a mature parvalbumin interneuron or a mature, something that looks like a layer 3 pyramidal cell or something. But I think we're going to get there. There are lots of people working on this. Now, this is combined with yet another remarkable technology, which is the ability to engineer genomes. So exploiting what's really the immune system of bacteria to hack up invading viruses from bacteriophages. There have been several new technologies. One is called, it's got a great nickname, CRISPR, which has now given us the ability to sort of take a standard set of stem cells and introduce one by one and then one by two and so on. Every mutation, we're just beginning to do this as a platform. We want to make these generally available, involved in autism and in schizophrenia. And then once we have these tools, the idea, now this is getting a little bit into the future, but I'm sure we can, together as a community, all do this, is differentiate these into the kinds of neurons we think we should be interested in. And then what's really important, because a lot of the mutations we're seeing early on in autism, and certainly big time in schizophrenia, involve synapses. And so the question is, can we make replicable preparations of synapses so that we can study small circuits in vitro and even use those to screen for drugs? And then we're all freezing down fibroblasts from people with autism, people with schizophrenia. Can we then, once we're really good at this, turn those into neurons and circuits and engineer out the genetic variations that we think are causative and see if we correct these phenotypes? Now, there are lots of holes in this already. We're not there, but it's very exciting to think that we can do this, because it would be awful if we ended up with a list of genes. This would not help anybody. We have to put them to work to understand autism and to lead to treatments. Now you might say, well, you're treating a whole person. You're not treating, an in vitro circuit. And I think the biggest challenge, and this will be coming from Kathy's end, will be to say, when we introduce these mutations, what do we see in these cells that is really germane to human beings? And I think this remains the biggest challenge. But if we can understand when we put in various mutations, the cells change, they change their firing rate, they change something, And we can then take patient fibroblasts with the same mutations, grow them into the same kind of circuits, correct the mutations, reverse the phenotype. Then the idea is that we could screen for small molecules that might, if we're lucky, have the same effect. Now, of course, in parallel, I hope there'll be great advances in behavioral interventions, but I think we need every tool at our disposal to do this. And then finally, yet another remarkable set of technologies that we really can't use yet, but down the road I think we can, are new technologies to understand brain circuits, which I'm not going to go into, but things like optogenetics and the new Clarity method for looking at brains, so that we can obviously look at greater levels of complexity. The other thing is, in every field of medicine now, there is a certain amount of disillusionment with mouse models based on the problems of evolution. So a perfect example is people die of overwhelming infections or sepsis because their immune system releases a storm of cytokines, of immune mediators, that causes capillaries to leak and organ failure. And for more than a decade, we've been able to cure mice who are models of sepsis. And not a single one of those drugs has been effective in humans. And finally, Ron Davis at Stanford went back and looked, and yes, humans have cytokine storm and mice have cytokine storm. It's only that the correlation between the two in terms of which cytokines, which receptors, was pretty close to zero. And so in many, many fields of medicine, cancer biology the same, people are moving toward cellular models, so maybe this will make what I'm saying sound a little bit less strange, as sort of the first place to be able to try to get a molecular handle on therapeutics, understanding that then obviously you have to go into whole living, if not animals, well, maybe we can debate this later, maybe earlier into humans, maybe we need human experimental biology. So to sum up, we have gone in a fairly short time from, I think, a really frustrating situation of not having a good toehold to begin to make progress. And then because of a variety of technologies, we are beginning to find genes. And I think schizophrenia is the farthest advanced right now because of the very large global consortia that have provided populations, but we're working to fix that for autism. In schizophrenia, there are now 110 genome-wide significant loci, and we've sequenced also the exomes of 5,000 people, and many of the variants that we're finding in exome sequencing actually map to genes already identified. So we have to get there in autism. And this is letting us then produce lists, but turning these lists into biology are very much reliant on these other technologies that I've described. And in some sense, this combination of genes and genome engineering and stem cell technology is just a year or two old. So I don't want to oversell it. I don't want to say that it's going to work for sure, but I am very, very excited about the possibilities of going forward. And that is exactly 15 minutes. So
let Kathy talk then. We'll
yeah, questions later. So
what?
You want to? Yeah, we want to do questions later.
Yeah.
And then.
But if you hold your questions until Kathy talks, then we'll. Unless you have a question of fact you want to clarify.
Rebuttal.
I should.
No. Yeah.
Okay.
Okay.
Perfectly clear. So you could probably guess Steve's career, especially his early career, was spent in molecular biology, dealing with opiates, addiction, and many other psychiatric and schizophrenia, and now is becoming more and more involved as the leader of one of the leaders at the Broad Institute in autism. So Kathy, can we go from cells to children?
Yep.
And the different approaches that you think are promising?
So is my microphone on? Yep, good.
Give your
Okay.
microphone.
Yep, I got it. Here it is. So I think I'm here for two roles. One is as a clinician, somebody who sees families and kids and adults with autism all the time. And the second is a clinical researcher and somebody who's interested in behavior, and particularly behavior of individuals and across development. And I think what I want to know is what changes across time, and then how can we for the better for kids and adults with autism and their families. In order to do that, we have to measure it. We can't just emote it. We have to figure out how do we know what's changing and in what context it occurs. I'm also, I think I have to confess, I am an autism person, so I do feel protective of families with autism, and I want things to get better for autism. And so that is a different perspective, I think, than a basic scientist who wants things to get better for everybody and wants us to accumulate knowledge. I'm less interested in knowledge than I am making things better. And I think that in autism, things have gotten better in the last 20 years. They're not miracles, but I think associated with primarily behavioral interventions, not just ABA, but behavioral things that families do, that schools do, that the communities do, that we do see things like more people go to school longer, academic achievement is better. There are more people in university. Now, that could be also that because we're diagnosing milder cases. But in addition to that, even from our longitudinal study, which we started 20 years ago, where most of the individuals have significant intellectual disabilities, they're now in their 20s, we see better adaptive behavior. People are more independent. We see better adaptive behavior than IQ in the people with intellectual disability, which we used to never see. And I think that's because people help. You know, the families, the schools, the community have helped these individuals. And I think in people with autism without intellectual disabilities, we see some people who are really doing incredibly well. And we see other people getting better all the time. So I think that we don't have miracles, but we do have progress. And there's a lot of data to show that we can teach or support people with autism to learn. Knowing that autism is a neurobiological disorder, I think has helped the field in a lot of ways. It has brought the attention of many very bright people. It's certainly provided support for families in the sense this is not something a family cannot make their child autistic. But we actually have had, I think, almost no significant, practical, useful outcomes of neurobiological data with all that time and effort and so many bright people. And I think my question, and I think Steve sort of led up to it, is how do we go from the things that he's talking about to the things that I want to see happen, and I think most people here do. We had huge hope, I mean, back in, I think, the early 80s, that if we found and described homogeneous populations, meaning classically autistic kids, and did genetics, we would find the gene, or the 3 genes, or the 9 genes for autism. And that didn't work at all. And so I think what's then happened is we come back and said, look, there's all these different genetic loci associated with autism. And then the hope, like in the Simon's SVIP, was to say, let's look at those, and maybe those will be the diagnoses. And then we'll move from there to treatment. The reality is the people within those genetic patterns are also quite different from each other. So there aren't simple answers. And I think what I want to sort of push for today is that we need to know more about behavior, not instead of basic science, but that we shouldn't let basic science push us to rule out or assume that we know everything about how people behave. I think deep down, I do believe there is something different about individuals who have very basic social deficits starting early on and repetitive behaviors and interests. And that when we look at the data, we don't really have a normal distribution of social behavior. We have 2 distributions. And part of the problem is that whenever we want to measure the severity of autism, We are so cut off that ordinary people don't even get on the charts. And if we want to measure social behavior in ordinary people, people with autism, the questions are almost not even relevant. And I think that gets lost often in neurobiological studies. People get in a hurry, they want to do something fast, they don't want to spend money on this because they want to do these really cool tools that Steve is talking about, and then we have a false impression of what's actually going on. The reality is that in human beings, social behaviors are incredibly related to context. That is, you don't have just eye contact out of the blue. You have eye contact in a situation. And what is part of the wonderful thing about human beings is that we are so flexible and we have multiple skills. And I thought an example was one time a young woman with autism came over to my house to return a sweater I'd lent her. And my son, who was about 9 months old, she came in and sat down and put her feet on our coffee table. My son was cruising around the coffee table and got to her feet, and he couldn't keep going. And so he pushed her feet a little bit. Then he tried to look in her face. Then he looked at my face like, what's with this woman? And finally, he threw himself over her feet, at which point she pulled her feet back and said, oh, what's he doing? My point is there at 9 months, he could communicate in 4 different ways that this is a really odd situation. Now, if he were 19 years old and her feet were in his way, he couldn't do those 4 things. Those wouldn't be appropriate. They would be totally different. Or if there was another situation, if my elderly mother had her feet up, then what I would expect him to do would be different. So my point is that those social behaviors are so tied to when and where and what we're doing and what are our expectations of each other. We have been able to quantify social deficits to some extent in context, but I think we could do a much better job than we can do right now. And whenever we start totaling up what somebody doesn't do or does do, we have to keep in mind what should they be doing or what are the alternatives. There was a really interesting article recently that said, are autistic traits autistic? And I think the reality is maybe not. I mean, sometimes, but there really isn't a lot of evidence that autistic traits and the kinds of simple measures that we often use differentiate autism from a million other disorders, especially combinations of things like low IQ, ADHD, anxiety. You add them up and you get poor social behavior. I think we also have to remember that parent reports, interviews, questionnaires are behavior too. They're not messages from God that are the truth. It's someone telling you right now how they perceived their child. And there are all sorts of factors. I mean, the most obvious ones are what the child's developmental level is, and then something that's changed tremendously in the last 20 years is access to services. So that suddenly, I mean, it used to be nobody wanted to have autism. Now many people think autism is far preferable to other diagnoses. And that changes, not intentionally, but it changes how parents are thinking about joint attention or about circumscribed interests. I think we're still talking about a very heterogeneous group. And even in what I see as the core group, it's not one thing. It's just that when we measure that core group, I think that we have to be careful not to just lump them in with other people. And you can make a normal distribution by having autism, people with low IQ, ADHD, and many other problems that can kind of fill in the middle, and then healthy development. It's not to say that dimensionalizing specific behaviors with or without autism isn't important. I mean, I think I just saw my 13-month-old grandson seeing a balloon going, it would be really nice to know, why is he doing that? And why is an autistic 10-year-old doing that? I mean, it doesn't matter who's doing it. It would be nice to know that. I mean, I don't know that that's tied to genes, but whatever it's tied to, it would be nice to know. It would be nice to know if the biting in Lesh-Nyhan can give us information about why some autistic kids bite their wrists. If the regression in Rett's, which is a different pattern, is similar to changes in the, and loss of skills in kids with autism, or if social difficulties in kids with ADHD share some phenomenon. So I think this importance of going across disorders is critical, but we also need to know who we're studying. I think one of the key assumptions in neurobiology, and you guys can correct me if I'm wrong, is that you want to get as close to the biology as possible. And I guess what I'm saying is that close to biology shouldn't always be away from behavior. Somehow close to biology, somehow biology and behavior in mice is considered more biological than behavior in people. We have better standards for how mice are treated in terms of when we study them, in terms of their sleep, in terms of the light, in terms of who touches them, in terms of than we do often with people. And I think that I'm not sure that that's good in terms of moving toward understanding what's going on in human beings. Again, I think a critical factor is somehow distinguishing what we're studying. And I think Steve was making the point that dimensions are critical, and I agree. But I think there's such an urge to dump a lot of things and call it a dimension. What is social behavior? It's not one thing. Just inherently, it involves somebody else. And so we need to know more about what that means. I think we need, if I had $1 billion, I would like a mobile way to measure brain function. Lying in an MRI scanner looking at faces is not necessarily the same thing as a social interaction. So I think the other issue is that science is very dependent on systematic studies that build on each other. Social science, I think, has been much less good about this than basic science. And one of the problems is that there's a finding here, and then people just head off on something else, and there's a finding there. And I think the problem is we link neurobiology, which has higher status, and then it links to social science without this systematicity, without saying, look, someone showed this doesn't work, or this measure does not mean what we think it does, but I'm not up on that literature, so I'm still going to link it to something else. And I feel like that's a place where our field has really been somewhat irresponsible. Overall, I think, in addition to the excitement of neurobiology, which we need, because behavioral methods are not going to they're not going to wipe out autism. I think behavioral methods can really help, especially kids who don't have intellectual disabilities, move forward, and kids who have intellectual disabilities can learn more, but we're not making the autism go away. So we do need that biology. But in the meantime, I feel like we don't want to neglect what actually happens to the whole human beings, not just for the clinical point, but even for the research. What are we trying to link these things to? We're not trying to link them to a parent interview or a questionnaire. We just use the titles of those things to have these links, and I don't think that's enough. So I think what we're interested in is just how do we combine these points of view. I'll stop there. Thank you.
Great.
You go in the middle.
I think we heard two very informed perspectives on how to approach something called autism. So I have a couple of questions, and I hope that you will as well. So Steve, how would you go from a gene to more complete understanding of how that relates to autism, or would you?
Well, I think the key thing is, if you ask me how do I go from a gene to a more complete understanding, the answer is you can't. The reason that we want to discover as many of the genetic, so let me just, we look for variants for mutations, for changes in DNA sequence that are associated with autism, because those point you to genes. We want to find genes because those point you to pathways, to the networks of proteins and RNAs that might control the postsynaptic side of an excitatory synapse. So what we want to do, or at least what I want to do, is to get the world, and when I'm feeling really frisky, I say to crush this problem, to get us, we'll never hit the asymptote, but if we could find 90% of the genes that are involved in autism, then we can start to put together this molecular jigsaw puzzle. Right now, we're in a place where we have a few percent and not the corner pieces of the jigsaw puzzle. But what will fall out will be molecular pathways, and those will help us identify something that we really don't know, which is which cells are important. And those will give us targets for, by knowing pathways, you don't actually have to necessarily perturb the system in one of the genes that has a variant that is in the causal chain. Once you know the pathway, you can begin to perturb it to understand the system and for therapies. My favorite example, because this is also abstract, is in lots of those of us with gray hair in the room are on a Lipitor-like drug, a statin. And the first clues to the importance of cholesterol actually came from observational studies from Framingham. But then the second set of clues was Brown and Goldstein discovered families that had such high cholesterol levels that people were having heart attacks in their 30s. And they used that to find ultimately a pathway, the pathway of cholesterol biosynthesis. Now, the medicines that we have target a protein that, HMG-CoA reductase, but they target a protein that is really has only a modest, if any, genetic effect on hypercholesterolemia. But once you find the pathway, then you can begin to say, where can I put my investigational fulcrum to perturb this pathway, to understand something, to perturb this pathway for a therapy. So I think you don't go from a gene. You go from alleles to find genes, genes to find pathways, and then pathways are a much better way into biology.
So Kathy, what would you gain from a genetic analysis? I mean, how would you subdivide a population, and how would you use that information? Do you care at all about these pathways?
Well, I think if we could understand how to get from pathways to behavior, then you would have some idea of what it is you're trying to change. I mean, with the idea that there would be some kind of medication or there's some kind of intervention. And I think that's
timing, maybe, when the genes come on.
Yes,
yeah.
That's right. So I think that's sort of where I'm pushing, is that I don't, I think I'm saying sort of like, well, I'm saying we don't even know exactly what the behavior is. We've learned a lot. I mean, we used to say autistic kids don't point, and now we say, it's not the pointing that autistic kids can't do, it's the pointing to draw someone else's attention. And then even beyond that, we've said, wait a minute, actually a lot of kids with autism have motor problems, and they're slower to use gestures. Some don't, but some do. So I think that gives us a better sense of what are we trying to change. I mean, not that we're linking genes to pointing, but just that we have a better sense of what it is we're trying to do.
I think we also have to be, I mean, I'm going to support your, there's no simple direct path from genes to behavior. One of the problems with the way people have analyzed transgenic mice is they've assumed I've put this gene in, I see this change in behavior, and this is causal. What we've learned actually from genetics is very humbling. There are a lot of shared genes across disorders, unshared as well. Kids with the same copy number variant or the same de novo mutation may look very different. In fact, some may be very impaired, but if you ascertain in a different way, others may be functioning quite well. And I think ultimately we're going to need an enormous amount of really good observation and really good ways of genotyping to begin to understand genotype-phenotype correlations with the understanding that there's a lot of distance between genes and behavior, right? We haven't talked about epigenetics and all kinds of other, right?
The back of my mind is to break down some of these traits, and I want to talk in a minute about intellectual disability.
Yep.
But when you say break down the behavior, my own view is that DSM-5 went quite far to trying to clarify some of the behavior. But I think that's been a very controversial topic. What do you think?
Well, I'm
Since you wrote it.
biased.
Huh?
I am biased, but I think we tried. I think that we tried to get away from little clusters that did have meaning to people, but had such different meaning to different people that they were confusing, and that they resulted in both clinical problems, but also certainly research problems, because you couldn't, one of the essence of research is to be able to replicate what you found. And if you don't know, if somebody has Asperger's as defined by this group or that group or this group, and you don't know who they are, then you're not going to be able to find it again. So I feel like that's a step in the right direction. I think what the unknown is like the 3 areas of social deficit we just made up. We have data, but the data came from the ADI, the SRS, the SEQ. You can only analyze what you've got, and we took what we had, and we didn't completely make it up, but we looked at the ways things were organized, the data that we had. So I think there's a long way to go in terms of improving that, and it's certainly also for repetitive behavior. So I think it's better, because I think having these tiny groups misses the point. But I think, to come back to it, I think we have to quantify dimensions, and I don't think we're very good at that yet. I think we aren't very, we can measure things, but there's still a huge temptation to glob stuff together that is really quite different.
But it'd be worth thinking about what would be improved measures. But Steve, what did you think about the DSM? And I know NIMH has taken off in a slightly different direction with trying to quantitate traits. What do you think about that?
So I think that the controversy notwithstanding, which I don't think was actually a scientific controversy, I think it had more to do with pride of authorship and so forth. I think that the DSM-5 change in autism was the only important scientific change in the whole volume. And the reason is, it's easy to make fun of the DSM. It's actually, early on, it would have been hard to do better, right? Because we didn't have any objective measures. And so smart people got together and tried to define behaviorally the core of these various syndromes. Two bad things happened along the way, one of them quite understandable, which is that the original framers of the DSM thought in terms of categories. I have a discontinuous, it's sort of like an infectious disease model. You either have smallpox or you don't have smallpox. And part of it is they were escaping psychoanalysis with all of its developmental continua, and they wanted to have real diseases. But it turns out, these heterogeneous polygenic common diseases, true also of hypertension, diabetes, by the way, are not very well captured as categories. They're much messier than that. Nature has not read our textbooks. The second problem was that the group that got together in the 1970s to write the DSM, the DSM-3, for reasons that are unclear, created an orgy of splitting. So they came up with an enormous number of narrow diagnostic silos. These have been very damaging to science, because to get a grant to write a paper, your patient population would have to fit into one of these silos. But these categories, I always call them, I figure I'm old and established and no one can really hurt me that badly. I call them fictive categories when I speak publicly. Meant that if you were to do an imaging study, you had to, someone had 5 of 9 DSM-4 criteria for depression. The problem with that is that some people are alike in only one category, and there are 151 different ways of getting it, and it never mapped onto anything anyway. So I think without blowing up the clinical enterprise, let's keep using it, clinically, because we don't have anything to replace it with. But I think recognizing that these are heterogeneous spectra and not making little boxes that control our thinking, and what Tom Insel and the NIMH are trying to do, we'll see how it comes out, but it's the right spirit. Liberate science. Don't undermine all of clinical care yet, but liberate scientists to think anew using different kinds of tools. I think that's very important.
I think where I would jump in is, it was really interesting. The beginning of DSM, we had these meetings, we're all going to have dimensions. But then the thing was, we can't measure anything because psychiatrists won't measure anything. So we're going to have dimensions, but we're not going to measure anything. And it was like, wait a minute, how can we do this? And I think that's part of my concern, is that I think the dimensions are fine, but we have to, first of all, kind of know, I think we should keep looking for what they are, because we don't know exactly.
Right.
And second of all, I think we do have to measure them. And I don't know that they can be measured in an interview or a questionnaire. We can create artificial dimensions.
Well, are you thinking about continuous automatic measurements?
Well, I think that in the end, maybe we can use technology. I think there's various things of looking at voices and looking at gestures and looking at facial expression. But I think we have to have multiple ways of looking at behavior.
So Steve, a lot of people have found, and I know you talked about it at the Broad meeting, that many of the genetic variants that are turning up in children on the spectrum with low verbal
with OIQ.
IQ. What do you make of that?
Well, so this is really the work of a number of people. I think Mark Daly and Elise Robinson, and we saw a very interesting paper.
We're finding that in the SSC.
In this direction from, we were just talking about it, but from Decode in Iceland. And I think what we're seeing is that if you have a de novo protein-destroying mutation in an important protein, I think in Elise, where Elise's looked, none of the children had IQs above 70, yeah. And this is why I was saying, without referring to this data, that we think that a lot of the core social deficits and repetitive behaviors and restricted interests are much more likely to be polygenic. In some sense, as disabling as they are milder aspects of the phenotype. And what I'm going to say is hardly ready for textbooks. It may be just completely wrong. It's so early. But I think what the lesson is emerging is that really severe mutations in proteins that you need to build the brain cause intellectual disability and often epilepsy, right? The brain just doesn't get built right from the beginning. And the deficits are, we don't have good, except for some of the work in inbred populations that Chris Walsh has done. We don't have any good genotype-phenotype correlations, but that's the idea. And that, but if you build a brain that has, clearly creates challenges, but also, can have a lot of not only effective functions, but in some cases remarkable intellectual gifts, then then that probably has to do with less severe tweaks to pathways that are involved in building the brain. And that's that's a heuristic right now, but that seems to be the picture that's emerging. So we need both, right? So one of the things early on, it drove me crazy. People prematurely declared it's all this, it's all that. This technology is good, this technology is bad. But it's empirical, right? And I think what we want to do is find as many of these de novos and protein damaging mutations, and we want to find and understand other mutations that are less severe in protein coding genes, and we want to find them all to put together this jigsaw puzzle.
Let me open it up to the audience. You've been very patient. Do you have questions? Yes. Short
Yes.
questions are better than long questions.
Oh.
There's a polygenic disorder, like most complex polygenic disorders, there are environmental factors. What are they? And secondly, in terms of quantifying those, are there any advances in biomarkers that can give you a number?
Yeah. So in some sense, the epidemiology of environmental factors the epidemiologists have been more in agreement in schizophrenia than in autism. But I'll give you an example of what's difficult and maybe where the genetics can actually help us find them. So there's a fairly well replicated finding that urban birth is a risk factor for schizophrenia. And you say, well, what is that a proxy for? What is causal in that? Urban birth. Being born in a city as opposed to, right? Well, it's almost more mysterious to know that than not to know that. And maybe by knowing something about genes and pathways, we can then go back to these environmental factors with some better, deeper hypotheses. But in some ways, the reason we're starting with genes, if you're just pragmatic, you want to know how these things happen, we're starting with genes because, frankly, we have technologies that can identify risk genes, whereas our ability to really incisively find the causal environmental factors is still lagging, but it's really going to be critical. Yeah.
New Jersey is
Yeah.
a risk factor for autism.
Wait, I'm a Jersey boy. Yeah.
So-
Yeah.
Martha, you got to talk into the microphone.
Just want to second what Kathy said about behavioral measurements. I have a couple specific comments. One is interested to hear you say that you feel really there are two groups, bimodal rather than normal, yet it is not showing up in measures. Perhaps one thing to do would be to study the interaction between the therapist and the child. If the therapist is forming that bimodal distribution, one maybe should study that interaction. The second point is I was very interested to note that the report issued by the Committee for the Brain Initiative, one of the points they made was the difficulty in gathering human subjects data. This was for neurology, for
Yeah,
DBS,
sure.
and the necessity to recognize and fund that sort of specific thing that the caregivers, the clinicians, the patients, they all are in this sort of social matrix. It's not quite a basic science setting, and it sort of has a special kind of need to be recognized and funded. Wanted to hear your comments about that.
Yeah, I agree. I mean, it's obviously, I don't know what I'm talking about, but I think it would
so
be.
obvious.
But I think, when we look at social behavior, it involves two people. And you can, lying in an MRI, conceive of, interacting with the picture that you're looking at. But that's quite a different process than actually being face-to-face with somebody doing something. And it's a limitation. I mean, even with the ADOS, we're talking about an interaction between an unfamiliar stranger in an office with a child. So all of it is moving far away from what the everyday social behaviors are. But that goes back to the whole idea of having to set a context. I do think you can see these two distributions in some data, but people tend to downplay it. And I think, again, if you jack up the number of people, if you include people, if you don't control for IQ, you don't control for age, and you don't control for general behavior problems, and probably to some degree parent stress, it's easy to fill in the bottom of a distribution that I think isn't autism and isn't even a social deficit like autism. It's not that autism stops here and everything else starts, but it's that there's something different, I think, about when you have those basic aspects of communication involved. And even as they get better, I think that affects relationships, which again can get better, but there are continua there that I think get lost in the shuffle of not being responsive, not, being nice. I mean, we just had someone today send an email about the ADOS saying, do I have to code crying if the child is crying because they're tired? And we're like, sure you do, because the reason why not because crying is diagnostic, but if the kid is crying, that's probably going to affect everything that they're doing in the ADOS, and you need to know it. So my point is, if you didn't code it, you can get a kid with a social deficit because they're miserable. And that doesn't help us. That doesn't help us understand what we want to treat. I mean, obviously we want to make people happier, but that's not the same thing as helping the social deficit in autism.
Many people.
Kathy, I wanted to ask you, you alluded to having a lot of money and doing some technology, so I wanted to probe that one level further. Do you think that ongoing active, constant EEG during social interactions, or a whole video classroom and, the really high power behavioral analysis of movement and facial gesture would be a step to go on, or you think we're not ready
I
for
think
that?
if we could do, I mean, I don't know enough about EEGs to know how useful that data is, but I think if there would be a way to do EEGs or continuous measurement during social interaction without interfering with the social interaction, I think it would be incredible. I mean, NEARS, I guess, does a little bit of that, is my understanding, but it's still, I mean, I think that's a direction we need to move in.
So if I could just take my reductionist tendencies, as you look at levels of complexity, often the observation at one level is somewhat unconstrained with respect to the level below, so that the same apparent behavior, like crying, if you didn't know the context, could have many different causes and probably different underlying neural circuits. If we could have some real-world continuous measures, EEG, whatever, that would help us, constrain the behavior and maybe distinguish different kinds of behaviors in a more objective way, I think that would be very, very helpful. And that's true all the way down, right? There are cellular processes that can be due to different molecular processes.
There's one aspect which, between the genes and the EEGs, there's the brain, but there's an increasing interest in what I'll call the mesoscopic level of analysis, where the Obama Brain Initiative is thinking of this, and we're thinking of it at the foundation in another context, of recording from suspect circuits multi-unit recordings during certain internal states, decision-making,
that'd be good.
fear, joy, navigation. And you wonder, that's not ready for prime time in people yet, but it may be it someday. But it's recording at the single unit level, but hundreds of them at one time, to try and predict these kind of internal states. Not signal average, but measure the flow of consciousness, as it were, whether that would bring us closer to an understanding of internals. If you just want to think of the ideal measurement.
But again, if you want to understand some aspects of behavior, one behavior can have multiple circuit strategies. A good example that would be intuitive is reading, right? Kids use very different strategies to read or to exert control over a prepotent stimulus. And I think we really want to be able to see those. And to Kathy's very important point, we want to see them in different contexts. You don't want to decontextualize the behavior.
But you want to make a measurement at the right level. Yes?
This is perhaps, I don't know, it's a question which you may not necessarily want to answer or even attack. But let's assume that there's some threshold of development after which, or prior to which you're not an autistic person, after which you are. Where in time or in the space of the development of the brain or in the organization of the genome do you think these thresholds would exist?
Well, it's an interesting question. I wouldn't want to leave it to just genes, because we know there are monozygotic twins, identical twins, who share all of their DNA, who are discordant for reaching a diagnostic threshold and for many other behaviors. So I think the threshold really is still has to be defined clinically. And I think in some sense we're going to, again, this goes to what I've called genotype-phenotype, but what genotype-phenotype correlations, including timing and, who's more impaired, who's less impaired, is going to show this gap, which Jerry's called the brain. But basically what it's going to do is going to define everything we don't know between certain genotypes and certain outcomes and help, I think, guide the investigation of stochastic, that is bad luck, real environmental interactors, all kinds of other processes in building the brain.
I thought the question was more,
What time? You mean how long?
yeah. I think the question was more
Yeah.
focused on, are there critical periods in the development of autism, which if we could identify, you could focus on.
Well, maybe that's better.
Yeah, I don't think they're critical periods, I guess. Well, I mean, sorry. I don't know about the brain critical periods, but I think that there are kids that move out of autism at different points in time. And I do think that happens. And it's arbitrary when you're in and out, but I think there continues, I mean, in our longitudinal study, there are people that at 20, had some vestiges of autism, although mild, and at 22 don't. So I think this is, I don't think that probably goes on forever, but I don't think there's a point where the whole thing about no language by 5, you're never going to talk. I don't think that anymore. You do know fluent, probably no language 1.5 or 4, and the chances of you being fluent. So is that what you're getting at, Bernie? Yeah.
Yes, absolutely. So a good example is actually, I've been thinking about it because David Hubel, one of the, along with Torsten Wiesel, discovered, among other things, the concepts that went into the idea of critical periods for development in the visual system. David died this week. But people really initially interpreted critical periods for binocular vision, for example, as a door was shut and that was it. Indeed, now I think people would say there are more gradients, right? The earlier you intervene, the better. But but these are processes that act over time. They have different timing in different circuits. Some circuits probably remain quite plastic. For example, I hope we all can still learn even as we become more mature. But a colleague of mine, Takao Hench, has found means to be working in the visual system to begin to reassert enhanced plasticity. Now, this is in mouse models, because being more open to change in response to environmental signals and then becoming less responsive in certain systems has biological controllers, and Takao and others are beginning to discover them. So it may be that, and again, this is still science fiction, but it may be that we might even be able to sort of create periods of enhanced plasticity, after the age of 5.
But then I want to ask Eric to chime in, but one aspect that Takao has pointed out was a deficit in GABAergic interneurons.
Right.
Really striking, at least in parvalbumin
Yeah.
staining neurons. And the question is there something wrong with the GABA mechanism, either the receptor, the chloride gradient, which is critical at certain periods of development in autistic children, and what the status of that literature is. And I raise it as a question. Maybe we can talk about it afterwards. We have refreshments, but before that, Eric London was going to summarize things from the point of view of a practicing doc. So, yeah.
Speaking to this now?
This one, yes. Okay, I feel a little bit intimidated being here and speaking with these people, but I'll get over it. I've been involved in the autism world since about 1995, and I still wince when I hear the word autism, and I think the reason I do is because I don't think I understand it. And even tonight, when people were talking about genes being autism, it doesn't make any sense to me. Kathy mentioned the social deficit in very young children and developing with social deficit. And if that's what autism is, then I think I understand that. But I'm not sure that's the way most people use autism, and so we have a problem. Steve talked about the DSM, and you should read his papers on the DSM. They're much better than what he said tonight. I think he's behaving himself tonight. But you know, he talked about some of the problems with the DSM, and some of them being the categories and the orgy of splitting, which is a nice way of putting it. Well, I think we have that problem with autism too. And I think the DSM-5 was moving in the right direction, but not enough. So here's the problem. What if the reason that progress in autism is not going well is not because of technology, but what if the problem is that we're not looking at the correct phenotype? And how do we correct that? The framers of the DSM knew that these were not real diseases, and they called them, I think, way stations or placeholders until we figure out what's really going on. But we've reified these diagnoses, and that's a word I didn't know till recently. Reified basically means we've made them real when they're really not real. These are just models. And that's a problem. So we think we know what autism is, we reify it. I bet you there are young students who haven't got a clue that there's even any virtue in debating the phenotype. I'll quote Dr. Rappin, who's sitting here today. She once wrote, "What makes autism so distinctive is that an experienced clinician or educator recognizes a classical case at a glance and rapidly suspects it, even in less severely affected person." At the same time, there's a quote which everyone in the autism world, I'm sure, has heard. If you've met one person with autism, you've met one person with autism. Lovas and Smith, in a 1989 article, said it very nicely. Autistic children initially give the impression of being a rather homogeneous group. Closer inspection reveals a wide range of individual differences, which cast doubt on whether the children really have much, if anything, in common. So there seems to be a gestalt, a kind of a feeling that we get when we see an autistic person. We kind of know it when we see it. And I believe that that's true, but I don't believe it's a disorder, and I don't believe it's a disease. I think it's more correctly should be conceptualized as a symptom. And whether even that symptom is fundamental to genetics or that's some sort of a manifestation of the brain's compensation mechanisms, I don't know. So our failure to find biological and genetic markers, we also have other problems which Steve did not talk about with the DSM, which he's written about, includes illnesses that cross diagnostic borders. There's a runaway situation with comorbidities, which I'd like to talk a little more about later. The indistinct boundaries between disease and natural variation, lack of predictive value of the DSM, lack of guidance for diagnosis and treatment, or even defining workup. So let me just talk a little bit about the comorbidities, which I think will illustrate the problem It's a clinical problem and then it's a research problem too. So if the demarcations of diagnosis which are made do not exist in nature, the probability that several diagnoses will have to be made in an individual case increases. And the whole concept of comorbidity never came up until the DSM-3 came out. So the numbers in psychiatry are just mind-blowing. 91% comorbidity for panic disorder, 91% for schizophrenia, 75% for depression. This is true in autism too. The ADAM network found 60% of autistic people had another diagnosis. 25% had 2 or more, and only 15 had no other diagnosis. Autism is comorbidly present in over 50 specific diagnoses. Autism is comorbid with nearly every organ system in the body. The Danish registry found that out of 16 types of medical symptoms, only 3 did not have an association with autism, and those were blood diseases, neoplasms, and genitourinary symptoms. Everything else was highly associated with autism. If you look at one more example, and I think this is a very important example, is in neurologic findings within autism, which have nothing to do with the phenotype, the diagnosis. DSM doesn't mention it. So one study found 95% of normal IQ autistics had minor neurologic problems. So 95% of high-functioning autistics had minor neurologic problems. So that's excluding severe autism, and that's excluding severe neurologic disease such as cerebral palsy. 87% had dysfunctional muscle tone, 39% had cranial nerve dysfunction, 75% had fine manipulative problems, 60% had discoordination and excessive motor movements, and 40%. So the question that I would ask is, does motor symptoms count for autism? Is that something we should have in the phenotype? When we're doing models, should we know this? It's a real problem. And so I'm real good at throwing stones, but it's a hard problem. But I will come up with a tentative solution in the next 2 minutes, which would be that, you know, we talk about looking at genes, and that's like really high power, that's electron microscopy. We talk about looking at what's in the office, and that's low power field. Maybe we have to even go lower powered and to look at autism as merely one of the developmental brain disorders. I think most people would agree that autism is a developmental brain disorder. And what's done in most of medicine, not psychiatry, is that you have the ability to put modifiers. So, for example, with diabetes, a person has diabetes with disorder of nephrology, with ophthalmologic problems, with ketoacidosis. And so you can describe something in a way that you have confidence is true, that this is a developmental brain disorder, and here are what we find. That way we wouldn't have the problem with comorbidities. That way we're able to, without prejudice, add what we see into that patient's chart, which would help the investigators, I think, because we would be basically telling them what the particular case has. And it would certainly be better for the patients. So I think I'll stop there.
Well, I'm going to suggest that we retreat and have some refreshments and continue the discussion with Eric, Kathy, and Steve. So thank you all very, very much.